Everyone. Hope everyone is doing very well. Welcome to Module 11 of the course. This is going to be sort of the last lecture where I will record the material for you. Next week is going to be fall break or the study break. And then next we will have few weeks. 3 more weeks to go. One is going to be the work week. There's 1 week. There's going to be the special topic week. So you'll be invited for a guest speaker talk, to give you an exposure to some ongoing research related to bioinformatics. So there will be a guest speaker, more information to come. So that is 1 week. Another week, which is work week. You're supposed, to be working more on your projects, and then going to be the final presentations week. So yeah, technically this is going to be the last module I'm recording, which is module 11. It's not a heavy loaded module yet. It's covering a very important topic and huge area of interest in research and daily life of many people who are struggling and fighting cancer. So this is module 11 about variants and cancer. In this module, we'll go over basically what is cancer, look into different aspects of that. This discussion in this module is not going to be comprehensive by no means. This is a big topic to be covered in one module. Try to cover some terms that are common in this kind of discussion. Make sure we know a little bit more about, germline and somatic kind of changes, allele frequency, trying to interpret gene next generation sequencing data, and so on and so forth. So hopefully as we finish this module, it's looking at the design of this whole course. I think this is coming more towards the application side. So we went through, general concepts right at the beginning of the course, more of a review of genomics, the history, and then went to the technology side, and then the bioinformatics tools, workflows and pipelines, which you have experimented with in the different activities, including the last activity which I've designed for you, where you had to go and run the Callisto program to do the alignment with real life data coming from Gene Expression Omnibus. You had to go and to download the GFF file, or that is the the annotation file from another database, and then try to do alignments, get the data set, go through a downstream analysis using one of the tools, and there we highlighted Network Analyst as an example. Now now you have all of these tools. So yes, it takes time to excel some of these skills, but you are well exposed to lots of concepts, lots of ideas. And if you have been doing your efforts, watching the lectures, doing your own reading and so on and so forth, working the activities and assignments, I believe we are in a good shape to move on. Where to, that's going to be related more maybe to your work, research, interest. Generally, we're looking into some application. So either you're going to have data coming from your own lab or data that you're interested to do analysis with. So this discussion, this module, I would say goes in line with, it's not that we're going to see everything coming together, but as an idea, as a concept here, cancer is where lots are applying these skills that we have went through in this study, meaning there are actually daily kind of work happening with in terms of producing, generating samples of data, cancer patients, genetic testing, and so on and so forth. So hopefully towards the end of this module, we'll be able to understand some types of, ideas such as somatic and germline mutations. We're going to look at the cell as well, some specific evolvement of cancers and how it may complicate the treatment. Yeah, and hopefully look more into some common terms like tumor suppressor genes, oncogenes, and so on and so forth. So cancer have been affecting widely lots of people. I mean, looking at the whole world, many people are actually affected, okay? Some actually are having access to the course of treatment or the plan of treatment. Some are not having that. Contributing to, this whole technology thing, this whole development, hopefully would make things more smooth, more accessible, less costly eventually. So we are all hoping that, cancer is the real humanity challenge, okay? I can say this. So if I'm looking at statistics for, it's about 30% of all. It's about 30% of all new female cancers each year. So that's in the States, okay? Yeah, thousands and thousands of, let me check here the numbers. So each year in the United States alone, about 240,000 cases of breast cancers are diagnosed in women, and about 2,100 in men, okay? And about 42,000 women pass away, die each year from breast cancer. So this is it is sad for everyone. This is only one country, okay? So if one would go and try to look into other countries, there are really huge percentages and numbers. So I gave just breast cancer as an example. That's very common. I personally know relatives, friends have, either going through this or had to go through this, keep hearing about it, other types of cancers. One of the struggles with cancer that we need to realize is symptoms. Usually the symptoms are not clear until the cancer is late in terms of the stage. Yeah, that's why there are recommendations. If woman turns to 40, they go and try to do things like mammogram to try to see if there are, any tumors there or so. Do some kind of consistent testing. And yeah, men also after 50, they will do some kind of tests as well. Yeah, so there will be some specific recommendations in some countries. I guess these are general recommendations, but it's different from, could be different from a place to another, but something that we certainly need to pay attention to and understand that it's a big challenge for all of us as humanity. It is one of the things I would wish that we had a similar approach to what happened in COVID. Sort of, there was a big competition between some countries. Eventually, there were, big knowledge and information sharing. So What I'm looking for is not a competition as compared to actually big knowledge sharing, information sharing, experience sharing. Of course, there are experience sharing in many throughout different centers and countries. But again, I think this approaching this with the ideology or the idea that we need to collaborate on something. Anyway, not to deviate with the discussion, it's something that we are really struggling with as humanity. We need to push all our efforts to, hopefully find a solution for this. So let's have a quick check-in and then ask, how are you doing? So let me know. Let's have this as the first treasure hunt question for this module. How are you doing? So how do you feel in this course so far? How are you doing in the semester? Any things you are struggling with? How are these weeks going on with you? What about the last, assignment? Okay, the Callisto one. I hope the instructions were sufficient for you to figure out. You were provided with scripts that will be very helpful for certain steps. So anyway, please share some feedback or something about how you are feeling nowadays. Of course, I would have loved to be in the classroom now and have some discussion, see what's going on with everyone here. Hopefully next week you're gonna relax a bit, in terms of the coursework, get the chance, to plan yourself, catch up if you have missed anything, any reading, trying to push a little bit more for the project and so on and so forth. This is, I think, let's see what's gonna after the break. I think of the work week, I'll try to invite teams for some meetings before the final presentation comes, or I would share some instructions. Let's see. What I'm hoping for is that you are really working on your own projects, and from some, I'm getting feeling that like I've asked one student, and the response was, "Okay, I'm doing the coding, I've done the reading." So it was very personal, kind of I and answer, not we. So what I'm hoping for is that there is a team effort in this project, not a single person. Make sure that everyone is contributing, because the evaluation would be reflected at the end by peer evaluation. We've already started this kind of discussion, which is about stages of cancers. When I say there are, the late stages of cancer, generally we're looking at 4 stages of cancer. When the cancer is in a small mass and localized, meaning it's not outside that specific organ, and we don't see any closed lymph node or adjacent tissues affected, then that's stage 1. So it depends on the type of cancer that we are referring at here. I think in breast cancer, we're looking at roughly 2 centimeters to maybe 2 centimeters to 5 centimeters, I guess. Okay, one would be very lucky if cancer is detected at stage 1. It's not that we don't have the technology, it's just a matter of knowing when to do the testing. Okay, who knows? And as I said, symptoms-wise, we don't see symptoms of cancer until it is really very late. Generally, we are speaking about stage 3 or stage 4. No question. This is when you start seeing or noticing some symptoms. Stage 2 is when the surrounding lymph nodes, and the lymph node is part of this immune system. Okay, so lymph nodes are very important unit in our biological system, and so when we have many of those, and when they are kind of the first respondents to what's happening with that specific organ. So, for example, think about the breast cancer. The kind of the closest lymph nodes adjacent one would be under the armpit, that area. So that one is going to be stage 2. Okay. Now, stage Well, maybe maybe these, I wouldn't say the armpit stage 2, let me say even closer to the breast, okay? Maybe the chest side or so. But the time it's under the armpit, that's when you start doubting, oh, this might be stage 3, okay? And the size of the tumor and so on and so forth. Now, whenever that specific tumor is, you start recognizing that there is another tissue in the body that is affected, that is stage 4. So, for example, I guess 30% or 3% of those who get breast cancer in women, the cancer is metastasized, which means it goes to another region in the body, and 33% of those where they get it is in the spinal cord. Okay, some may end up getting it in another place or organ. And by the way, the breast cancer, although it's very common and many are affected, and it's the focus of so much of the cancer treatment efforts, it's perhaps not as severe as brain cancer, as liver cancer, and so on and so forth. But anyway, so the staging is very important, and usually this is done by taking a biopsy. And one of the things to note about the biopsy is, I'm not the physician in this thing, but it seems that taking a biopsy is something one needs to be careful, meaning to hopefully you're going to start the process of treatment soon. So I don't know if this question is will address that, is taking a biopsy, how much is going to affect the spread of the cancer. But I've heard patients saying that after the biopsy, they felt that the cancer is growing. So, getting the needle or something there, pushing these cells there could really spread them somehow. But anyway, that is a needed procedure to take some biopsy and start looking at the tissue. It takes generally maybe roughly 2 weeks to get back with what is specifically that type of cancer or stage. Here's an example of a stage 3 or 4 melanoma. And again, I like that it's saying 3 or 4. You hear this from physicians. So sometimes they say, well, we are in doubt if this is stage 3 or 4 until we get, because they do different types of scans. Like this scan that you see here. This is PET and CT scan. So usually there is what's called, they will take a CT scan, so try to scan the body, try to recognize any spots are there, and they will do bone scan to see if anything is metastasized to the bone side, and so on and so forth. And if anything is felt, because these technologies again have certain limitations, but if there were any kind of hint that there is something there, then the physician would forward you to PET scan. PET scan is more accurate, or MRI scan. Okay, it's going to be generally, okay. This gets tricky, and I'm going to mention this on the next one of the next slides. So even when they see nothing, it doesn't mean that there's actually no cell that went into other position in the body. That is stage 4. Stage 4 means, some of the cells or one of the cells sneaked out of this position, like the breast, and went to another location. So here we are looking at melanoma, skin kind of cancer, and then metastasized, meaning that there are other places, right? There's this area for the adrenal, and there's this one for the gastric. So this is kind of a PET scan visualization showing how this got into different areas. So some of the statistics used there in the research to indicate what's called the survival rate. So what is the percentage if I get, or the chance if I get cancer to live? Now, by no means this is conclusive. So I'm going to share this because it's common but really it's very case dependent, and I will say why. But I can observe from this that female breast cancer, this is telling us there are huge success. Prostate cancer, which is common in men, especially, you know, after 51, is highly recommended to go and do some tests and examination. And this is kind of telling you the different types of diseases and how they are difficult in terms of treatment. So success rate maybe in female breast is higher than lung, and so on and so forth. Yet it really depends on the stage. We know for sure if it is localized, that is stage 1 and stage 2, which is again, you are lucky. Like I hope you get no disease. When I'm saying you, I mean the person, okay, is lucky to know that they are in stage 1 or 2. So if it is stage 1 or stage 2, yeah, you know, this is kind of still even a little bit, you know, in the surrounding region, there's a high chance of survival. Okay, high chance of survival. Procedures would be chemotherapy, you know, sometimes for the breast, there would be lumpectomy or mastectomy, which is removing part of the breast or the whole breast, depending, and then radiation therapy, and depending on the type of the cancer, which they do early testing. One of the first things they would do to decide to look into what is the type of the cancer. Is it hormone like estrogen positive and progesterone positive, and HER2 positive or negative, and all of these kind of metrics indicate what type or how difficult is this cancer in terms of treatment. Many of those who get the general type of cancer or the common one, they are having at least a positive, two positive, meaning estrogen and progesterone positive, which means the cancer cells are, you know, getting their energy from the estrogen side. So if we block this estrogen, we can control this. And so anyway, there will be treatment. So we go chemotherapy, we go surgery, we go radiation therapy, and then hormone therapy. Along the way, we may have immunotherapy. Something not common to hear about in this whole treatment protocol or procedure is dietary and emotional, and many of the things which are also important to be developed. It's not the physicians are not experts in the dietary aspect, but you as an educated person or patient, you know that you have to have a full approach to that, because you know it depends on certain details, like what about sugar intake, how it's affecting the cell, and so on and so forth. What about controlling that? Is it going to help in fighting these cancer cells? Just one example. What about the you know, reducing the consumption of whatever products that are around us where there are some chance of carcinogenic ingredients or compounds. So this figure is telling us, if I want to read it as is, that female breast with localized female breast cancer, the survival rate is 98% for 5 years, which means the chance that you survive 5 years and live beyond that is almost, you know, 98%, almost guaranteed. I would say all like guaranteed, almost guaranteed. Okay, see the big drop when this is distant, we're talking about 30%. But again, this is not to generalize those cases. I'm spending a little bit time here with some details that might not seem really bioinformatics intense, but I guess that is very important, guys. If you are watching this, you know, if, yeah, as I said, I had like a person very close to me, and that person got that type of cancer. So I know what they went through. So I guess educating yourself well about this would be very helpful. Okay, very helpful. So anyway, why it depends on the one person or in specific cases as another treasure hunt question. So although I can use this as a reference for studies, but what would you think some of the thoughts or some of the reasons why this cannot be easily generalized for every single case? Okay. Share your thoughts on the second treasure hunt question. Some of the things that I would like to say is that it really depends on the type of the cancer, even if it's distant, okay, even if it's distant. How's the treatment progress and how that person is responding to the treatment. And sometimes we know that after some time there is a natural developed resistance for certain treatment courses through these cancer cells. So the conditions would really matter, the details would really matter, okay? So it is common nowadays to hear about people with metastatic cancer who lived, we hear these stories. I talk to physicians and they tell me, we know patients who have been living more than 10 years with metastatic, which is distant cancer, okay? So again, these are average number, are not conclusive. They may change with the time. What we are all hoping for is all of these, are gonna be pushed here, okay? And that's what we are all hoping for. Tumor heterogeneity is a natural development of this discussion I'm building with you. Why? Because there is some of the challenges when we look at, tumor and cancer. This is, we're having normal cells, something is going to take over here, and so we start having a tumor population of cells. And they are in between these normal cells, and they start growing. So we get, split, divide, the cell is going to divide. Well, we're talking about the cancer cell now, so we get into tumor population, second tumor population, and keep going. Okay. So when we're looking at this, I can refer to what's called the type of mutations, the somatic mutations. There is some mutation here indicated by these blue kind of curves or ticks or specks. These are the original driver mutations. What we're meaning here by somatic mutations, in the body we have two types of mutations. Some that can be attributed to the cell, to the egg or sperm, affecting the time of conception, and so that is what's called germline mutation. Okay, somatic mutations. So what happens, is a kind of mutation that indicate mutations in the cell after that stage. So sometimes these mutations are not necessarily inherited. Sometimes they are, result or coming from some exposure, from some things going on in the environment, and so on and so forth. One of the things also that I would like to say, do not misunderstand this and think all cancer patients are having some kind of mutation. I'm going to repeat this. Do not misunderstand and think necessarily all cancer patients that they should have some sort of mutation. Nope. Okay. So this is going to answer directly a question that why we don't use early detection. We can. Okay. And so if you have that specific gene mutation, you may decide to go to surgery at an early age, and that's it. Get rid of where that location, might be, get hit with the disease. Maybe you're going to just do systematically, like every few months you will do a mammogram, or you will do some kind of medical imaging, try to diagnose what is the chance of having a tumor. But in many of the cases, there are no specific mutations. However, it's also worth to note the following. What we are seeing here, and specifically in the this area of clonal mutations, these are genetic changes that occur in the early stages of cancer development. Okay, and they are present in all cancer cells with a tumor, and they had a common ancestor. These cells were the common ancestors. So now we have mutations that are there in all this population of cancer cells. That's why we call them clonal mutations. and they're kind of considered the driver mutations. Okay. So some of the types, like breast cancer genes, related genes like BRCA1 or 2, are often clonal because they provide critical growth advantages to the cell. So this kind of, growth advantage to the cell, this is what would refer to the type or link it to the type of the mutation. As compared to the subclonal mutations, these ones to come later, okay? It exists in a subset of cancer cells, and these types of mutations vary widely across different regions of the same tumor, or between what we call primary tumor, which is the main organ where the tumor was present, and the metastasis, that is the other organ or distant location, to see this type of cells. They indicate some heterogeneity, and this kind of heterogeneity, because we're seeing now, recognizing some different mutations as compared to the previous ones. This could, if we end up seeing different kind of mutations, this is going to complicate certainly the treatment. So genetic testing at some stage early in the treatment is very important to inform us, are we dealing with the disease? Like, this is one of the factors, again, when I'm looking at these statistics, I wouldn't say conclusive, because this is just looking at stages of diagnosis. What about the types of mutations? Those would certainly be important factors. So someone who's not having any mutation is certainly at much lower chance of having a recurrence of that disease. So if we want to put that tumor evolution perspective and try to monitor the cell development and the course of treatment, for example, we have this illustration here. Okay, so in this this is called fish. It's called the fish plot, showing the branching evolution of a single tumor. So an interesting question would be, if you are looking at this sphere, and when I point this out as as also a treasure hunt question, when was treatment administered to this patient? So what we are having here is that we are having the cell growth with certain mutations or variations. So we're indicating 5 of them in this plot here. And these mutations could happen because of several reasons: environmental factors, random errors, replication errors, any different reasons or causes could be there. Okay, so which we have time point T1, time point T2, and time point T3. So which one you would say is, in terms of subsequent time points, is the one where treatment was administered to the patient? Okay. One hint, if you start thinking about this, indicating the kind of the shell or the cell size or shape, and so on and so forth. Okay. Now, when we're looking at this, okay, these kind of mutations might be the mutations that are going to affect the immune system. So again, depending on what types specifically of mutations that we are dealing with, T1 would be the time point that the treatment was administered, because we start seeing shifting, especially that this area is almost going away. Okay, but the question, another question would be, did it work? Well, we'll say partially, because the green and purple subclones eventually were eliminated. Okay, but still we have other areas indicating that is red and yellow subclones. Okay, so some subclones were removed or went away because of that treatment, but some subclones stayed, okay? And they were responsive somehow. And the term that would indicate for this phenomena in the evolutionary context is that a founder event, okay? Eventually, this what's happening here. Because a founder event is the event that occurs when a new population is established. So a new population of cells established from a small number of individuals coming from a larger ancestral population. Okay. So these kinds of changes that would indicate how tumor is evolving, eventually could really lead to treatment resistance. So it is documented that chemotherapy, for example, after some time, it could have negative effects. Some also, depending on the case and the person, they might not be as responsive anymore as then they had the beginning of the treatment. In other words, you would hear stories that, I got this type of chemotherapy, the first session, second session, third session, I started realizing the tumor is going down. And then we moved with another chemotherapy. And that's why they are using nowadays more of what's called combination therapy, multiple drugs. Because if you use one, first you may go with all the side effects of that. Other thing is maybe you're not at a higher chance of, developing resistance to that specific drug. So you would hear some physicians, well, we exposed that patient to this kind of chemotherapy, but we didn't, after a few cycles, we didn't see any progress. So we stopped, we shifted to another one, and so on and so forth. Metastatic seeding, it can, that's, sneaking to other parts, affecting immune system. Vascularization is blood. Cells need blood vessels to feed them. That's going to be the power generator. So when tumor cells start developing this somehow or getting closer to these blood resources, it's going to really empower them and they make the spread much easier. So after that intro about cancer, what as bioinformaticians, if we have a case that we want to explore further, so let's say one patient, so we certainly can use computational methods to document and generate a report, and part of that is visualizing the data that we have. So here we are revisiting the IGV genome viewer from NCBI, which is very, it's a very common and way as an integrative genomics viewer. It's a common tool, for visualizing this type of data. So let's go through integrative genomics viewer.org. As I mentioned, it's it's by the Broad Institute. Okay, this tool, not NCPI, sorry for that. They have a few options, by the way. You can download this at a desktop. I would highly advise this time you go and download this and have it there as one of your tools. The tool is there to visualize the genomic data. So IGV viewer is having some reference genomes. Sorry, guys. IGV is listing some reference genomes that you can directly use. You can upload your own genome. By now, it actually can give you the option to directly download from their servers. So you have already kind of default list of options that you can directly choose, or you can get from their servers if it's one of the species, or you can upload your own. And this is going to be the reference genome feature side. And then here we're kind of going to map different tracks. Generally, you can think of these tracks as different files. We're going to line up on top of the genome and try to see the different, the differences there in a visual kind of inspection. Maybe I'm interested in specific gene, and I want to clearly see, this would be the perfect tool if you want to see which mutation is specifically there, which nucleotide is changed, and so on and so forth. And here we can be dealing with a set of alignment sequences from the patients, like in this case, and where the bases are and from the reads are aligned. These are the alignments of the reads, and then this is the reference genome. And so bases that do not match the reference sequence are having highlighted colors. So this is going to make it easier for us to zoom in and look into the quality of this, and so on and so forth. Okay, so we can be looking at two types of groups. Okay, so you can add as many tracks you want to make the comparison. Like here we have, there's tumor sample and normal sample, and then try to make some comparisons. Why having both would be helpful, because then we can address some questions. Okay, so some of these questions that we are interested in. Oops, I'm having some. Oops. Okay, anyway, so some of these things that we're looking at is knowing when we're comparing, for example, tumor and normal samples, if this is kind of germline variants or somatic variants. So the question is here for us is, are these variants germline or somatic? How can we answer this from looking at the browser here? And one hint, I want to remind you that we have two groups of samples. So I'm going to leave this, or, I want you to think and give me this as a treasure hunt question. Think about it. What germline means and what somatic means. Okay? What germline means and what somatic means. Okay? Mutations. And again, somatic mutations arise during an individual's lifetime, as compared to something that happens and early in the stages of germ cells, and as I mentioned earlier, at the stage of because of the DNA, inherited DNA in the egg and sperm, and so on and so forth. Somatic is more during the lifetime. So if you keep this in mind, what do you think? How can we tell, I'm not pointing at a specific variant, but how can we tell from the IGV browser and looking at two types of groups if these are germline or somatic? So this is one treasure hunt question. And the answer is going to be, well, if what we are looking at here is they are present only in tumor samples, like the changes or mutations or variants as compared to the reference genome. So maybe there is some area here, and I see this. There are changes here, or maybe sorry, there are changes here in this area compared to the reference genome, which is only part of the tumor area. Okay, or the tumor sample. This might indicate that these are present only in tumor sample, these changes. So this is kind of somatic variant, because these somatic variants will be only visible in tumor samples, versus germline variants, which will be changes that we recognize. Maybe there are some change here, some change here, maybe linked to this. So it's in both. So when reference to here, I'm just pointing at the gaps here. Doesn't mean any mutation, okay? Perhaps I should have used a different way, but because this would be colored actually, okay? So maybe I would use this. There's some indication of some change here as compared to the original one, since it's all in both samples, that germline. If it is in one of them, okay, this is going to be more of, if it is in the tumor sample, somatic one, okay? And also similarly, if I'm looking at the homozygous variants, okay, I'm going also ask you to think a little bit about it, okay? Because homozygous, we're looking at two variants, two versions of the gene that we call one of them allele. So these variant alleles related to that gene as two versions of that gene, one coming from each parent. If they are the same, that's homozygous variant. If they're different, that's heterozygous, right? So how do you know which one is which based on IGV viewer? Well, if almost all the reads at a particular position will show the variant allele, so let's say maybe I'm at a particular position, okay, that's homozygous. Why? Because all the reads at that particular position will show that variant. And possibly we can focus only on one patient, okay? We don't need to go through two patients, okay? So we're focusing on one. Similarly, if only half of the reads will show that variant, so maybe I will see here some variants here and half of the reads, okay? And the other half, there's nothing, okay? As compared to the reference, then this would indicate, one copy of the gene has the variant, while the other copy does not have it, okay? So variants in cancer, we'll be discussing this more in the next section, but before I stop here, there are certainly many types of variants that cause the types of cancers that we have. Amplifications, deletions, a different driver, insertions, deletions, germline, and so on and so forth. And this kind of shows the drivers by variant type for different types of cancer diseases. So I'm looking at the breast here. This seems the higher area, which is missense, okay, as compared to mesothelioma. This is here is more of a deletion, and so on and so forth. So there are documented studies about these different types of genes. So I'm going to stop sharing the screen here, because what I want to do is actually open the IGV viewer and go through some quickly. I have a window here. Where's my window? Oops. Okay. Seems I've closed my window, so I'm opening another one again. And oops, here we go. And this is the new tab. Okay, cool. So what I'm gonna do here, okay, guys, so I'm going to visit first igv.gov. I think this is where we igv.org. Sorry, not gov. Okay, sorry. Okay. So you have the desktop version, you have the web app, and I think this is kind of, I'm not sure about this, probably some maybe web visualization. So this is the full tool here. There's also JavaScript, which means it can be integrated to your online web tool. So I'm looking to go with the web tool here. And the moment you are here, by default, it's loading the human genome 38 for us. We can choose, one of the chromosomes. You see here the chromosome, so we can go to the X chromosome and then start, you can select a region, start zooming in, and the directions indicating, and oh, sorry, this is where we have the annotations of the gene names and so on and so forth. Okay, let me see. Yeah, and this is from RefSec genes, and so we can keep going. There is no annotation in this area, so it's probably an uncoding area. You can still see the sequences. Okay, let me go here. You can come here and choose a chromosome and then indicate where you want to go, like specific position up to which position, or you can indicate specific gene and you would see it here. Okay, see this? So we have here BRC. Okay, and this is where getting to the sequence. Okay, these colors indicate the different nucleotides. So you will see this once we are deep into this. So thymine with red, adenine with green, and cytosine with blue, and guanine with, I think this is orange. Okay, so as you zoom in and out, this is what we are getting. So this is basically choosing the genome. You have a list of genomes that you can go through. Okay, and you can upload your own and so on and so forth. Now, what I want to do is let's go to the default view here. What I'm going to do is I want to upload some sample files. So that you don't feel distracted and where I get this, I'm going to refer you to a nice website. This is bioinformatics.ca. Okay, where they run very nice bioinformatics workshops. .github.io, and this is from 2021, module 2, lab cancer. So you go to this one here, and it's very nice tutorial for cancer analytics and alignments in integrative genomics viewer. Okay, so if you are interested to further look into this tool, I would really suggest that you can go and play with this here. It should be very helpful. They also give access to the normal sample and tumor sample. So I'm going to do this upload here because we have only the reference genome. So I will use tracks for that because I want to add a new track. So I'll say I have a local file, and I have, perhaps you don't see this, but I have now my Finder window where I need to upload 2 files. So I would choose the samples, and I have to upload 2 files at once. So I have to select 2. In this case, the BAM file, which is the binary alignment file mapping, and the BAI, which is the index file of that alignment. So any alignment output, because we're going to put not in random reads, not just reads. I want to put the aligned reads, okay? Otherwise, it's not going to be helpful for our viewer here. So I'll say open, and that is get me, you have here normal BAM, as you see in the viewer. I'm going to add another track, local file, and I will use the tumor sample, tumor alignment output. So this is what I see here, okay? Maybe the alignment reads, because these are small files, okay, that you have here, are not full, so that's why we have an empty and saying, zoom into some specific location to start seeing something. So we can do this, okay, and there are many options that you can go through, but let me quickly jump here into, for example, chromosome 9 at the specific locations here, and click on search. So this is what we start seeing. Very interesting alignment reads and tops of that specific RefSec gene, FUBP3, and we start to look, compare both reads, aligned reads from both samples and try to see with our eyes if we are able to inspect or see if there are any issues, any things of interest to us, and so on and so forth. Maybe you're interested in that specific gene or that specific area, so we can zoom in a little bit more and then, you know, try to find what interesting things we are having. Okay. Yeah. And you see here, this is the actual reference genome C, where we have C, the main track, right? But here we have some changes, right? And you can see some changes are common in both patients, some may not. Okay, so this is kind of the game here. Try to search for those cases and spend more time. And so the exercise will take you through different locations, and let's try this one here. Yeah, this is interesting, right? You have this gap through this whole thing. That's why it's a homozygous deletion. The deletion is there everywhere in all the samples and reads as compared to the original genome. So it's a very nice tool to spend time with, explore the different options, and so on and so forth. So that's it for this part, and we'll continue in the next one.